Okay. Hi, I'm Dr. Vergano. We're launching attempts to record little bits of information that you guys might find helpful for your children and your families and your providers as you navigate your diagnosis of Coffin-Siris. A lot of the times you get, your test results explained to you, but in the moment, it's sometimes hard to process everything and you don't have all the time in the world with your genetic counselor or geneticist. So we're going to just go through some basic ways that you can have genetic testing and maybe what that might show you. And hopefully you can come back to this and look at it again if you guys have questions. So, genetic testing has come a long way in the last forever, 50 years. But it still can be pretty confusing, especially if you don't have a science background or if you're overwhelmed by your child's diagnosis or if your child is sick. Sometimes we just don't process these things. So, um, in general, I would say that most people nowadays who get a diagnosis of Coffin-Siris have either had something called a microarray. Sometimes it's called a SNP. SNP stands for single nucleotide polymorphism. More commonly, we just call it a SNP microarray or a microarray. Or people have had broader testing, which is sometimes called whole exome sequencing or whole genome sequencing. Those are two different things that we'll talk about. So, if you think back to high school biology, you probably vaguely remember seeing pictures, big pictures of chromosomes, right? And that everybody has a certain number of chromosomes within their body. You may have learned about certain conditions where individuals have additional chromosomes, such as Down syndrome, or a missing chromosome, such as Turner syndrome. And you may remember learning about something called a karyotype, which is a picture of all an individual's chromosomes. So that's a big picture look at all of your DNA that's been packaged into these little components. You may have heard me in the past liken genetics and genetic testing to a book. So if you envision your book or your child's book as their instructions on how to be made and how to function, um, a karyotype would be the chapters in the book, so big collections of information. A microarray is kind of like a karyotype in that it tells you whether there's DNA there or not there, but it zooms in at a much higher resolution and tells you about small missing or extra pieces of DNA within those chromosomes. So again, with the book analogy, it would be a bit like seeing that part of a sentence is missing or some of the letters of a word are missing, right? Not big chapters, but smaller areas. And sometimes children who have Coffin-Siris that's diagnosed from a microarray, you may have a finding that says something like you have a small missing area on chromosome 6, and the exact location is Q25, and that involves the ARID1B gene, and therefore your child has a condition consistent with ARID1B related Coffin-Siris syndrome. Or it may tell you things like there is an extra piece of DNA on a chromosome, and we don't think that's impacting your child at all because all of us have missing and extra pieces of DNA. It's what makes us different as humans, and it doesn't necessarily always confer disease, right? So there may be missing or extra pieces in DNA that are passed down through families and have absolutely no clinical consequence whatsoever. It's just part of who we are, and it doesn't necessarily mean that it's bad or that there's something medical associated with it. Okay. Whole exome and genome sequencing really came about clinically probably within the last 10 years, I would say. Exome sequencing came about first, and more recently, we have the ability to do clinical genome sequencing. An exome is a test that looks at all the protein coding regions of our DNA, which are called exons, EXONS. And again, to the book analogy, the way I explain it to parents is exons are words in a sentence that give the sentence meaning. So on this slide, if you look at any of the words, if you took out things like or for or those are words that don't necessarily give the sentence meaning because if you took those away, you and I would probably still understand what the sentence meant. So exome sequencing only looks at kind of important words. So it would look at test, it would look at looks, it would look at typos, it would look at misspellings, it would look at genes, um, such that the reader still has kind of a sense of what the author was getting at. If you think about all of our DNA as an apple, I really like analogies if it's not become abundantly obvious. If you think of all of our DNA as an apple, exons really represent a very small amount of our DNA, a very thin slice of the apple. There's a lot more to our DNA than just exons. We don't know all about what changes in the other parts of our DNA fully mean. But genome sequencing will look at everything. So it will look at the exons. It will look at things called introns, which you might liken to the words that we initially took out. So like for, those are kind of like filler words. But we've learned over time that some conditions can have changes in those words and they confer disease. So genome sequencing looks at the whole apple. Exome sequencing looks at a small slice of it. But that small slice contains a lot of meaningful information to kind of how our body works. And you may have done this test with your child, so they may have asked for a sample from you and your partner, or the child's other parent, to sort of do comparisons. So it's not looking at all your DNA necessarily, but it's looking at it kind of comparatively to what they find in your child. So if they find a change in your child, they will look and say, did this change, is this change also seen in mom? Is this change also seen in dad? Or is it new in your child? So part of the difficulty, I think, for non-medical people, which is probably the majority of the world, in understanding test results is that you can have a change in a gene and it doesn't necessarily mean it's causing disease. Most people are familiar with the word gene mutation, and that word has been used for centuries to describe a change in a gene that causes disease. The trend now really is sort of to shift the narrative away from kind of stigmatizing language in medicine and genetics. And so we no longer call things mutations. We might refer to them as pathogenic variants. And all that means is a change in a gene that causes disease. I think you've probably all heard me talk about the fish in The Simpsons with like the 12 eyes. I think that's what most people think of when they think of mutants, right? It's something bad and not meant to be and was a mistake, right? None of which describe humans or creatures, right? So we've kind of changed the language to represent a more objective view of changes in DNA. There is a spectrum of ways in which a lab can interpret a test result. So they may look at a change in a gene and say, wow, this is a really significant typo in this word. We don't know what this word was meant to be, and we can't understand the rest of the sentence. That might be what they're calling a pathogenic variant. So disease causing, we've seen it in other children, say with Coffin-Siris, and we know it causes Coffin-Siris. There are other changes in genes or words that may be a typo, but may be able to still impart understanding about what the word is. And the example you've probably heard me use a lot is the word theater. So in America, it's spelled T-H-E-A-R. Sorry, T-H-E, how do I spell things? Theater. But in England or the UK, they spell it Theatre, right? Those last two letters are flipped. It's the same word, same meaning, and they're both acceptable spellings. So that would be on the other spectrum of variant interpretation. We would call that benign, meaning it's a change in the word. It does not cause disease. It's not doing anything to your body. And somewhere in between there, I may get this in the next slide. Let me just check. Well, forgive me for going back a sec, but can I go back? Yeah, okay, I'll get to that in a second. Um, you may find something in your report called a variant of uncertain significance, and I think we'll talk about that. But basically, what that means is that's kind of the middle of this spectrum. We found a change in a gene. We don't know yet whether or not it's causing your child's features, and we don't know yet whether it's associated with a disease. So we can't fully give you a definitive diagnosis. Okay. I'm going to change gears a little bit and talk about what you may see on your child's test results. So if you have those handy and you want to look at them while we're talking, you can see a little bit of what I'm referring to. So there may be parts of the test that describe the gene that your child has a change in, and it will often say it's, for example, the ARID1B gene. This is associated with ARID1B related Coffin-Siris syndrome, and that's the diagnosis. There may be a section that says mode of inheritance. And what that means is not who your child inherited this change from, although that's what you would think it means, but it means that theoretically, what's known about this gene, when it's passed down, do you need one copy of the gene change or do you need two copies of the gene change to have the condition? So it may say things like autosomal dominant or autosomal recessive or X-linked dominant or X-linked recessive. The vast majority, in fact, all of the CSS genes are autosomal dominant, meaning that doesn't necessarily mean your child inherited it from someone, but if they were to pass it down, they would have a 50% chance of passing that gene change down because only one gene needs to be passed down to have the condition. Okay, so that's more like kind of a theoretical thing about the gene itself. There should be somewhere else if you submitted parental samples where it says inherited from. And that's telling you in your child's case, this specific case, did your child inherit this change from one or both of their parents, depending on what the gene is? This is different than what we were just talking about with mode of inheritance. So this is specifically pertaining to your child's case. This is not a theoretical scenario. The overwhelming majority of Coffin-Siris variants are what's called de novo. It's Latin meaning from new. It means it arose spontaneously in your child, was not something that they inherited from you or their other parent. Um, and was, and even if they did inherit it, right, none of these things are things that you can control. Nothing is anyone's fault. You didn't do anything wrong. You did not do anything. So it's just to give you a sense of where this came from, right? And we can talk maybe in another talk about um, how gene changes occur or inheritance. But um, while there are a few cases of Coffin-Siris syndrome where they are inherited, it's pretty unusual for there to be a typical parent, meaning like no learning or developmental differences, who has this gene change, passes down the gene change, and their child has a more significant clinical picture. We can get into that another time, but that's just sort of to give you a sense of that. Probably should have switched these slides, but this is going back to what I was discussing regarding the spectrum of variant interpretation. So at the bottom here, you'll see it ranges from pathogenic, which we said was disease causing, all the way to benign, which we know means does not cause disease. So in your child's test report, you may see specifically next to the gene something that looks like this that begins with a C dot. And that just refers to copy DNA. That's just the way it's shortened. And the 1, 2, 3, 4 is like a location. So it's telling you where in that word or where in that gene that typo occurred. Everything has a location. And so that helps us understand where in the gene your child had a change. You may see also a letter and then one of these like greater than signs and another letter. This means that the typo or the misspelling resulted in one letter being substituted for another. So instead of a G, which should be there, right? There's an A. So A replaces the G. And labs use a number of different ways to figure out whether a gene change is pathogenic or benign, or whether it's this variant of uncertain significance. And we can talk about that maybe in another clip as well. So I'll say it's very confusing. Sometimes it's even confusing for providers. But if you have questions, genetic counselors and geneticists are there even after your appointment. And it's important to understand that we don't expect you to understand absolutely everything that we went over if it's just one time. And we like being able to be there for families to explain things. And you've heard me say a million times that, genetic testing doesn't define your child. It doesn't indicate their worth as an individual, but having a better understanding of kind of what makes your child who they are and looking at the results and the science behind it helps some families to understand things a little bit better. So thanks. Hopefully we'll do more of these.